Welcome to our session. The title of our discussion is Navigating Strategies for Comparative Effectiveness from Trials to Real-World Evidence. My name is Preeti Jingran, and with me I have Craig and Alexandra, or Ali. We all are affiliated with a healthcare consulting organization called Genesis Research Group. And we specialize in real-world evidence, HEOR, access, and pricing. So my job here is to help you understand the importance of this topic, as well as why plan early. I think most people in this room probably know why comparative evidence is critical, especially in terms of making access and reimbursement decisions. So access decision makers use comparative evidence to understand the value add of a product coming into the market. So it's always relative, incremental evidence or incremental value, both from a safety and efficacy perspective. What really becomes really important in that situation is to understand what is your comparator, in which population, and based on which endpoint. The holy grail when it comes to comparative evidence is to have head-to-head data from clinical trials. But in many instances, you are not able to conduct head-to-head trials to generate comparative evidence. In that instance, knowing that we still have to generate comparative data to support your payer and HTA submissions, there are other methodologies and approaches which are available to generate this evidence. And that is where indirect treatment comparisons and external controls come in. Now, in our observation within pharma as well as in consulting, what we see is that we don't often start having these conversations about the strategy around comparative evidence early enough. These discussions are very nuanced, and you have to take into consideration the evidence base, which is available to determine the fit-for-purpose strategy for your comparative evidence. You also have to spend a lot of time with your cross-functional matrix partners within the organization to de-risk your strategy, as well as work with external experts, because, like I said, that these methodologies are extremely nuanced. And lastly, you would want to understand the evidence or the estimates which are generated out of these comparative evidence. What is the impact of that on downstream tools and resources which are used to support access and pricing decisions, such as your cost effectiveness model, value narratives, etc. So as you can see that there's a lot of moving parts here. The scientific rigor and discipline and the nuanced approach you use to generate this comparative evidence requires early planning, a lot of critical thinking, and the experts in the room to really inform your decisions. So hence, we wanted to take some time to really focus on what are some nuances with indirect treatment comparisons, as well as external control arms, and why start early versus using this as a tactic for late planning. So with that, I'm going to actually transition to Craig, who's going to focus on indirect treatment comparisons, and later on, Ali is going to come in and talk about external controls. And lastly, I will close the session and talk about the key takeaways.
So I'm going to introduce you to some of the methods for indirect treatment comparisons that can support your comparative evidence plans. I want to start with just recognizing that there's a broad world of different ITC methods, and we're going to focus in on two of the most commonly used, the network meta-analysis, or NMA, and matching adjusted indirect comparisons, or MAICs. For MAICs, we're going to talk about two different versions, the unanchored and the anchored versions. So digging into this, really your choice of ITCs depend on the available data sources and several different assumptions that are important to make distinct inferences about comparative effectiveness. I'll walk through each of these, starting with NMA. This is a method that allows you to make multiple comparisons against multiple treatments. There's no requirement for individual patient level data. You use aggregate data that's generally in the published literature for pivotal trials. And it requires that your evidence base be connected via a common comparator. So if you look at the diagram here, we have treatments A through E, and they are all connected through treatment A. And this allows us to create comparisons for example, D versus B or E versus C. If we move into the center, we have anchored MAIC in the center and unanchored MAIC in the far right here. The difference between these two is the need for having a required common comparator. So if you look at the anchored MAIC, they have the common comparator A still, and you compare B versus C through that. In the unanchored version, we no longer have that common comparator, and in this case we're comparing B to C directly. You might wonder, why don't we just do unanchored MAIC all the time? The answer to that is relatively simple. It requires much stronger assumptions that I'll get into next. Looking at both the MAICs, these are most often leveraged for comparing just your trial to a single competitor. So you generally use your inpatient level data and then adjust it to match that of the comparator trial. The anchored, as I mentioned, requires that connected network, and the unanchored does not. If we start digging into the assumptions now, I'm going to spend my time staying at a higher level. So if you've heard about things like transitivity, constancy, consistency, this is what I'm talking about here, but I'm trying to put it at a level that's approachable. So in order to make inference that is valid, you, for each of these methods, must meet certain criteria or understand the limitations that are associated with that. The first one is homogeneity within trials. So if you have multiple trials within a treatment, those should be relatively similar across all of those populations. Similarity across populations means that if you have multiple trials across different treatments, hypothetically, you should be able to have patients be in each of those individual trials or be eligible for those. So the idea that they can be exchangeable between any of those trials. Next is consistency of direct and indirect evidence. Direct evidence is that direct head-to-head evidence that happens in RCT. Indirect are those that are estimated via these networks. And those should be consistent when you're doing these types of methods in order to make valid inference. Lastly, the constancy of a treatment effect refers to that same treatment effect should be constant across all the trials that are under study. In the anchored and unanchored versions, these are called conditional constancy of relative effects and conditional constancy of absolute effects, because we're conducting adjustment that basically allows us to say we meet that condition. So moving forward, if we think about the limitations and risks associated with each of these different methods, I want to acknowledge that under ideal circumstances in a perfect world, we would likely get the same answer, and that the assumptions are very important to making that valid inference, because this rarely ever happens in practice. If we look at each of these different scenarios, some kind of pragmatic ways to think about this, the chances for a wrong answer increase when you have different populations, so that first assumption's kind of not valid anymore. If your studies are conducted differently, so endpoints are measured differently or designed differently, or you have different study ways of conducting your studies in terms of the specific design. And ultimately, what you need to do as practitioners is understand how much variation there is in these assumptions and what that impact is for your inference when you're making it. I do want to point out that for anchored MAIC and unanchored MAIC, because we conduct adjustment in those scenarios, we are able to somewhat attenuate that risk of increasing error, but only under certain circumstances. So for unanchored MAIC, there's a very strong assumption that you have measured all possible treatment effect modifiers and prognostic factors that could impact the treatment effect estimates. In anchored MAIC, that's relaxed a little bit more, where you need to make sure that you do measure all of your treatment effect modifiers and not have large variation in prognostic factors. Those are important to consider when you're thinking about moving forward with each of these, which I'll talk about on the next slide. So what are the important questions to ask before proceeding with an ITC? First, let me talk about best practices. We should always conduct a systematic literature review to identify potential comparator studies in an unbiased way. The unbiased way is extremely important here because we want to make sure that we can show in our submissions that we did not pick and choose which studies to include, and the criteria should be pre-specified and made sure that it's clear from the starting point. Once you conduct that, you can assess your evidence base for the feasibility of these different ITCs, guided most importantly by the assumptions of each of the methods, so the things that I've just discussed. Once you have this broad view of feasibility, you can tie it back to your strategy and understanding what your strategic needs are, because there may be scenarios where you might choose, for example, MAIC versus NMA, even if you might have a pretty strong network. Okay, that is really dependent on the situation, and you need to work with your experts in-house, stakeholders who have that knowledge, like statisticians. So when we move forward, we always should be thinking about what are our key comparators, what's important to our strategy, what does that body of evidence look like, and how is it connected, and what are the differences between what are the potential variations in those assumptions, and how would that impact what our conclusions are. In a general framework, for my best recommended guidance, NMAs are great for multiple comparators of interest when you have a large body of connected randomized evidence in populations that meet those criteria that I talked about, right? So if you have a lot of variation in your populations, you're going to introduce that error in the conclusions. So typically these are rapidly expanding or crowded treatment landscapes. For MAICs, anchored MAICs are generally preferred over unanchored MAICs. It's a very rare scenario where unanchored MAICs are preferred or accepted, so always keep that in mind because of those additional strong assumptions, and any of your submissions will certainly be scrutinized over those choices that you make. So MAIC is generally best suited for single comparators in small networks that are not suitable for NMA, or when that question supports that. When we have that variation in the populations where adjustment is required, and this generally falls into sparse treatment landscapes, things like rare disease or single standard of care settings. Next, I'm going to pass it over to Allie to discuss external control arms.
As we've heard from Craig, both NMA and MAIC require a connected network of existing randomized control trials. When that evidence network has a gap, that's where external control arms, or ECAs, come into play. Unlike MAIC, which will use patient characteristics to adjust for differences between trials, an ECA aims to replace a missing control arm entirely, using data from real-world patients. This is particularly relevant in a single-arm trial where an RCT may have been deemed unethical or infeasible. ECAs require fit-for-purpose data from sources typically like EHRs or disease registries. Several analytic methods exist, with some requiring individual patient-level data from both the trial and the external data source as well. The strategic value of ECAs is most clear in instances where they're used to contextualize findings from a single-arm trial with real-world data comparators. And I'm going to walk us through an example to help bring this to life. In this scenario of a rare indication, the pivotal trial won't be able to generate comparative data, so it's essential to choose another strategy. Proactive data landscaping and monitoring of the evidence base in parallel with trial design ideally is important. We always recommend a structured and documented approach to data source selection, including a clear assessment of fit-for-purpose criteria for each candidate data source that's considered. Here, a disease registry is identified that captures patient-level data on key prognostic variables and clinical outcomes taken from routine clinical care, making it a viable external comparator data source. When designing the ECA, a transparent, pre-specified, and stepwise approach goes a long way towards building credibility with stakeholders. Frameworks like target trial emulation can be used to help us work through some of the trickier concepts, like aligning time zero and endpoints across the trial and external data. The most important thing I hope that you'll take away from this example is that a well-conducted ECA should be an explicit part of your evidence strategy from early development, ideally developed in parallel with trial planning. Building off of the slide that Craig shared, considering when each approach may be best for NMA and MAIC and expanding that to external comparators. In general, ECAs are best suited to scenarios where the standard of care is the appropriate comparator, where there's no connected evidence network, and when a real-world population can closely mirror the patients that are enrolled in your trial. Because of these different conditions, we most often see ACAs being used in oncology and rare diseases, though it's certainly possible to execute this as a strategy in other TAs under the right circumstances. Successful execution of all of these strategies, ECAs included, requires buy-in from both internal and external stakeholders. As external stakeholders, HTAs and payers will often hone in on the hardest to verify assumption that's present in your study, whether that's focused on data quality, patient selection, or outcome ascertainment. So bringing a lens of rigor to any of these approaches is very critical. And with that, I'll turn it back to Preeti to share some closing remarks.
All right. Okay. So as you could tell from the presentations you heard from Craig and Ali, there's a lot of moving parts and a nuanced approach to really determining what is the methodology you should be implementing when it comes to indirect treatment comparisons as well as external control arms. And so scientific rigor and discipline is absolutely critical because that could make or break your analyses. Most of the HTAs have very detailed guidelines in terms of what they accept and how they recommend you approach a comparative evidence generation. Therefore, we're recommending plan early, de-risk your strategy, implement the broad evidence strategy around comparative effectiveness into your integrated plans. Build it into your development plans much early in the drug development process. Never compromise on the methodology. And essentially, if there are three things you want to walk away with is plan early, align early, and focus on the scientific rigor, which is absolutely critical to generate this comparative evidence. With that, for the first time, we're finishing our HEOR theater on time. Usually we always go over. So we have enough time to take questions. I think we still have to continue to wear our headphones, right? Is that correct? That's how we listen to the okay. All right, so if you have any questions, you can come to the mic here in front. And we're happy to answer the questions you may have around methodology, strategy, any of the elements around the topics we just discussed. Questions?
I'm happy to at least get one question that I think might be on people's mind answered, which is when you're faced with a decision between, let's say, unanchored MAIC or conducting an ECA, which one might you choose? And I think that really becomes about trade-offs and considering a lot of what I had already talked about, these assumptions of how, for example, a unanchored MAIC, how that comparator trial meets those assumptions compared to conducting an ECA where you are in a non-trial setting and you have many other biases that come upon us, such as immortal time, outcome as specification, and so forth. So really, it's about making a judgment call as to whether or not what trade-offs you want to make and which one you think is the most valid to estimate that comparative effectiveness data.
Other questions? Yeah, go ahead. Please come to the mic.
Hi, Stephen Crawford from Takeda. This is not so much a methods question, but it's a question a lot of us are grappling with in the pharma space, especially over the coming years as we approach, joint clinical assessment, and some of which are already ongoing now. In that sort of future, you're not dealing with running 5 ITCs. You're running potentially hundreds or thousands of ITCs. So how do you sort of take this kind of methodology and extrapolate that to having to do it hundreds and hundreds of times?
Yeah.
And I know we're in an AI world, and that could become our future path for NMAs, but we're not there yet.
Yeah, I mean, even at the conference here, there are like softwares now coming up with where you can do NMAs. So time is to tell, I guess, in terms of how this space is going to evolve. You're absolutely right. You do have to grapple with multiple comparators across multiple different countries, and therefore you might land up with dozens of ITCs,
And hundreds of picos.
hundreds of picos, yeah, and that's something we've been grappling with already in terms of generating all these picos and prioritizing it based on input from the country affiliates as well as JCA. I don't have the crystal ball here, honestly, in terms of exactly how you would approach these multiple ITCs, but I would see if maybe Craig or Ali have anything to add here.
Yeah, I think, yes, the chance for automation is there. I think we do need to talk about whether that's the right decision or not, because you can do automation, but I think that increases your risk for error.
Oh, yeah.
Right? And so it's how do we do it thoughtfully? And I'm going to admit I don't have the right answer, but how do we do it thoughtfully so that we can make sure that we're not drawing the wrong conclusions, because ultimately, patients suffer and things like that. So it's going to be a struggle, and I think understanding how to optimize that workflow while still respecting, I'm a statistician, respecting the assumptions that go into these things so that we can make valid inferences is going to be a real challenge in an increasingly AI world.
Yeah.
Thank you.
I think we'll all be connected on this topic, right? Because we'll all be watching it.
Hello, my name is Dan from Arginex. So I had a question. So I think there's a lot of sensitivity to present any sort of comparative data, particularly from a reg standpoint. So just wondering, how common is it that this type of data is able to be presented proactively by companies?
Proactively by companies to support the launch or commercialization of the product is what you mean?
I mean, that or any other particular instance, not necessarily just for a launch, but just in general.
Yeah. So I think, again, it depends is the answer. There's a lot of nuance to what can be presented proactively versus not. And you're absolutely right that there is a lot of sensitivity around comparative evidence. And that is why it requires a lot of alignment within the organization, because you have to de-risk not just from a regulatory perspective, but also from a commercialization perspective in terms of what data should be or could be presented. Now, as far as proactive dissemination is concerned, there are some new regulations from the FDA where if the population, the indication is consistent to the label, then you could present the information proactively. So again, lots of layers, lots of moving parts, but nonetheless, there are possibilities of presenting this information proactively if it makes sense.
Are you referring to CFL?
Yeah, I'm referring to CFL, exactly. But CFL in itself is a wild card right now. Nobody really knows what's in and what's out of CFL. So I think in the next couple of years, it'll be much more clearer in terms of what could be covered under CFL and what would not be covered under CFL.
Thank you.
Yeah. Other questions? Ali, do you want to add anything from an external control arm perspective?
Sure. So it's kind of in my world of ECAs, I see them being used both for regulators and for payers, but my background is a little bit more in the regulatory space. And when you've already sort of cleared the hurdle with regulators to move forward in a single-arm trial design, at least from an external comparator perspective, there can be a little bit more of an opportunity to be proactive in bringing forward a external comparator strategy. I think the bar continues to be extremely high from a regulatory perspective, especially around fit-for-purpose data. I think the methodology are sort of there, but the data still have some sort of gaps that need to be addressed within our space to see better uptake and acceptance of these strategies.
We can take one more question. No? Okay, well, we want to leave you with just three key takeaways. As you can see, there's a lot of moving parts here, so start thinking about your evidence strategy when it comes to comparative evidence much earlier in the drug development process. And, work with the experts in the space when it comes to the methodology, because again, like I said earlier, these are very nuanced approaches and require a lot of critical thinking and de-risking of the strategy within the organization as well. With that, we want to thank you and enjoy the rest of the conference.